stage 4 disease. However, up to a quarter of these patients suffer from irreversible hearing loss, which is a known complication of treatment. So given how hearing loss in a relatively young population can significantly affect their quality of life, we hope to study the potential therapeutic options which could perhaps one day be used to ameliorate this. So in our study, we hypothesized that dexamethasone and D-methionine protect hearing through different mechanisms, and that administering them together can provide an additive benefit. I'm sure most of us are familiar with dexamethasone. In vitro studies have shown dexamethasone to reduce apoptosis in hair cells exposed to either cisplatin or radiation. In addition, intratympanic dexamethasone has been shown to reduce cisplatin-induced hearing loss in randomized controlled trials. D-methionine, on the other hand, is an essential amino acid. It has been shown to reduce cisplatin hearing loss when administered through the round window in animals and when given systemically in humans. So for this study, we used organ of Corti cells from mice. The cells were pre-incubated for 3 hours with either dexamethasone, D-methionine, or a combination of both. These cells were then exposed to either a control, cisplatin, radiation, or both cisplatin and radiation. We then measured cytotoxicity and cell viability using various assays. We also looked at the mechanisms of actions of dexamethasone and D-methionine using immunoblot analysis. So in our study, we found that dexamethasone was able to protect hair cells against both cisplatin and radiation. D-methionine was on the other hand only able to protect against cisplatin. When combined, an additive benefit was observed. So looking at the fluorescent images on the left, the green specks denotes dead cells stained by the cell-tox screen cytotoxicity assay. The group that was exposed to no treatment, denoted by NT, has the most amount of fluorescence as expected. Less fluorescence was seen in groups exposed to dexamethasone and D-methionine, while the least amount of fluorescence was seen in the group exposed to both drugs. So looking at the bar chart here, sorry, my background's a little noisy. Looking at the bar charts here, these further illustrates the cell toxicity and cell viability of the various groups of cells exposed to various treatments. For the chart on the left illustrating cell toxicity, the vertical axis represents fluorescence, which is the measure of cell toxicity. So the shorter the chart, the better, as it represents less cell toxicity. For the chart on the right, the vertical axis represents cell viability. So the longer or taller the chart, the better. The charts illustrate that cisplatin-induced cell death could be partially inhibited by dexamethasone alone, whereas fully inhibited by D-methionine alone and a combination of both drugs. On the other hand, radiation-induced cell death could be attenuated by dexamethasone alone and the combination, but not D-methionine alone. So looking at this diagram here, our study also looked at the mechanisms by which dexamethasone and D-methionine acted to provide autoprotection. Cisplatin, which is indicated by the red arrows, and radiation, indicated by the blue arrows, can trigger p53 activation, resulting in apoptosis activation. Alternatively, they can also activate inflammation, which exacerbate ototoxicity. Using in vitro ototoxicity model, we demonstrated that D-methionine could fully protect against cisplatin-induced apoptosis via blocking p53 activation. On the other hand, dexamethasone exhibited protective effects against cisplatin and radiation treatment by attenuating p53 activation and upregulating BCL-XL expression to halt apoptosis activation, as well as inhibition of the ERK1/2, NF-kappa B activation, and COX-2 expression to ameliorate inflammation. So, in conclusion, our study found that combining dexamethasone and D-methionine exhibits additive protection against ototoxicity from cisplatin and radiation therapy. And dexamethasone and D-methionine have distinctive mechanisms to protect ototoxicity in the process of apoptosis and inflammation. Thank you.